Alright guys, so the last video we covered ended off with this slide, and I said that we were going to continue through this topic of cell communication by studying what is known as the signal transduction pathway. Now that signal transduction pathway is basically a way for either cells to communicate with one another or the outside environment to communicate with the inside of the cell. Okay? And the signal transduction pathway can actually be broken up into three distinct phases: reception, transduction, and response. And so the video I'm producing today is discussion of the reception phase of the signal transduction pathway. Okay? Now, in the reception phase, a signal molecule binds to a receptor, causing it to change shape. That is the impetus for the entire thing. That is where this entire thing gets kicked off, where you can have some kind of chemical signal from the outside environment relay some maybe relatively complex message to the inside of the cell, specifically usually at the nucleus. Okay? Now, at its most basic, at your most basic understanding of reception, what you have is the binding of a signal molecule to a receptor. Okay, that signal molecule is called a ligand or a ligand, however you choose to pronounce it. I've heard it both ways. I generally do ligand. Okay, but that ligand is going to interact with a proteinaceous or a protein-based receptor. And that interaction is highly specific. We have highly specifically shaped receptors that will bind only to specifically shaped ligands or signal molecules. In this fashion, we can make a signal or respond to a signal that is highly specific. I use this term specific a lot for a reason, because there is this specificity that is needed to get the correct message across. Okay? Now, once this signal molecule interacts with this protein-based receptor, there is generally a conformational change. Okay? This term conformation or conformational means shape change. Okay? There's a change in the shape of the receptor. And oftentimes we can consider this the initial transduction of the signal. Okay, I'm going to back out of this presentation real quick here, because this term transduction, I've mentioned it a couple of times just in passing, and I think it bears going over a definition of what is transduction, or what does it mean to transduce something, because we're going to use this term a lot. So I went to the internet where you go to find these things, and I went to see if I find the definition of to transduce, or transduction is a form of this word. To transduce means to convert into another form, or to cause or undergo. Okay? And that's effectively what we're doing in this process. We are changing things. We're taking a signal and we're transforming it or transducing it, changing it into something else that our cells can actually understand. And when they can finally understand this signal, they will then respond to this signal. Okay? So most of these signal receptors that I've been talking about that interact with these ligand molecules are embedded in the plasma membrane. However, some of these signal receptors can actually be found inside the cells themselves. And there's a reason for that. These are called intracellular receptors, and they are ultimately found in the cytosol, or even sometimes the nucleus of a target cell. Okay? Now, the reason we have these intracellular receptors where most of the other receptors on the surface of the cell are on the surface of the cell, the reason we have these intracellular or inside of the cell receptors is because some of the chemical messages that our cells respond to can be hydrophobic or very, very, very small chemical messages. Okay? And what have we talked about in terms of very, very, very small or hydrophobic chemical messengers? They can pass right through the cell membrane. They don't need to interact with anything that happens to be on the surface. And the reason that they can pass right through is because they're hydrophobic or very, 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 very super small. Okay? If you remember the cell membrane, we just got done with this in chapter 7, right? The cell membrane is highly nonpolar. It is highly hydrophobic. And if you have a chemical or a compound messenger, a chemical messenger, that needs to send a message to the cell, and if that chemical message is hydrophobic, it can just go right through. So you don't need to interact with anything on the surface. So examples of these things would be things like steroids, okay? And so when you get one of these chemical messages that can just go right through the cell, what it's ultimately going to do here is interact, let me get the pointer here, is interact with one of these intracellular receptors. So this purple thing right here that almost looks like a tooth, like a jagged tooth kind of, is the intracellular receptor. The signal molecule under study here is a hormone molecule, testosterone. So it's lipid-based, it's fatty, okay? It's nonpolar. It's very highly nonpolar. It'll just diffuse right into the cell, okay, right across the cell membrane. And then it will interact with a receptor protein. And this is to ensure that there is some, again, specificity in where this message is going to be sent and what this message is ultimately going to have as a response, okay? And so we see once this hormone molecule diffuses into the cell, it interacts with the receptor protein and creates what they call a hormone receptor complex. That's nothing you necessarily have to memorize, but it's good to know that that's what they call it, okay? Once this complex has been created, it then enters the nucleus, okay, and it binds to the DNA. Now, this is a very specific response that is dedicated to testosterone, okay? This happens to be what testosterone does once it goes into the cell and relays its message. It binds to this protein thing, and then this complex goes into the nuclei, and then the nuclei, then the DNA that's inside the nucleus is transcribed, okay? And then we make a protein. So that is the specific cellular response for this particular guy here. Now, different cells are going to have different interactions to different proteins or different signals, okay? So keep that in mind, that this is just one very, very specific response. There are many, many different types of responses and interactions and relays of molecules, okay? But I wanted to show you here the intracellular receptor and how it works. I'm going to get this one out of the way because majority of the other ones are surface level receptors, okay? These receptors are found in the plasma membrane, and they work with water-based molecules or water-soluble molecules. And these water-soluble molecules need some kind of receptor on the surface of the cell because they can't actually diffuse into the cell. So they need to interact with something on the outside of the cell, okay? So these little water-soluble molecules interact with these cell membrane surface receptors, okay? Cell membrane surface receptors. These are protein receptors found on the surface of the cell, okay? Now, there are three main types of surface membrane receptors. They are called the G protein linked receptors, the receptor tyrosine kinases, and ion channel receptors. Now, we're going to go into each one of these and talk about how they work specifically and how they work to receive a message and what is the first step of that reception, okay? So we're going to see a lot of pictures here. We're going to explain a lot of pictures, all right? The first one that you see right here is a picture of how the G protein linked receptor works, okay? Like I just mentioned, the G protein linked receptor is found in the plasma membrane. If we see up in the upper left-hand corner, this is a picture of what it would look like, you know, in like a 3D space filling model. Okay, that's kind of all the alpha helixes and stuff like that that are embedded in the cell membrane. Okay, and that's what it would really look like. The picture we see down here, this big purple blob, that's the idealized version. That's the one that you're going to probably try to remember for a test. And you try to memorize, visualize that picture and say, all right, I got that picture in my head. I know how this thing works now. Okay, so it is a plasma membrane receptor that works with the help of something called G protein. Okay, that's where we get this term G protein linked receptor. Okay, this is the receptor called the G protein linked receptor, and it works with the help of this green thing right here called G protein. Okay, now G protein acts as an on-off switch. Okay, it can be activated. And this is something that a common theme that we're going to see throughout this material. Okay, we're going to see molecules becoming activated. All right, and one of the things that you have to watch out for when you see these molecules becoming activated are two things. Okay, oftentimes you will see in these pictures, you will see the shape of the molecule change. Okay, and you will also see a glowing like yellow aura. Okay, that indicates to you that a molecule has become activated. And when a molecule has become activated, that molecule is on. And then when that molecule is on, it can then go activate something else or turn something else on. Okay, that's how these things work. And so spend time understanding what I just said. Okay, when a molecule is active, it means it's on. But not only does it mean it's on, it means it can go activate something else. All right, that's a very important point to make, and we're going to see it here in G protein linked receptor. Okay, so now remember, we're talking about how this signal molecule interacts with the receptor to receive the initial message. Okay, that's what we're trying to do here, and we're going to try to relay this information in every one of these protein receptors that we talk about. Okay, so how does G protein linked receptor work? All right, one of the first things we see here, this is the G protein linked receptor complex up in the upper left hand corner, picture number 1. Okay, that's the whole complex, that's everything involved. All right, so we see G protein linked receptor all by himself over here, and it's not active. How do we know that? We know that because it's not a glowing yellow molecule. Okay, we see G protein sitting here all by himself, inactive, because it's not yellow, it's not glowing yellow. Okay. All right, the next picture we see something has changed. Okay, one of the first things we notice is that the little orange signal molecule, the ligand, has bound into the what they call the active site of the receptor. It's a very specifically shaped site that will only fit this very specifically shaped molecule, right? Okay. Now, one thing we notice here is that there is a change in G protein linked receptor from picture to picture. So if you look at this molecule, this purple molecule here, and you compare it to this molecule here, you will notice that there is a distinct change. Let me pull up my drawing tool here and see if I can get you something that helps with that. If you notice right here, kind of bowls out like that, kind of bulbs out like that. And if we look at the same portion right here, it kind of goes concave. I don't know if you know, but that is a change in shape. So that tells us that G protein linked receptor, this guy, went from inactive to active. Okay? So one of the things that makes us understand that the G protein linked receptor is active is it's, number one, it's changed its shape. What else do we see? What else do we see? The glowing yellow aura on the outside. I'm using a yellow highlighter on something that's already yellow so you can't actually see it. All right. So all around that thing there's like a little yellow aura, okay? And it surrounds all of G protein linked receptor and helps us understand, yes, this is active. Okay? It's an active molecule now. All right. So now we've got an active G protein linked receptor. That's a good thing because that starts off everything else in the signal transduction pathway. Okay? Now the next step in the transduction of this molecule or of this pathway is the activation of G protein. Okay? And G protein is this guy right here. Right now it's not active, and over here we notice that it is active. Why? Because we see it's glowing yellow. Okay? And the reason it has been activated is because it has interacted with another molecule that has already been activated. Right? G protein linked receptor right here has been activated, and it in turn will then activate G protein. Okay? And so that one right there is also active. Okay? Now, if you remember, I said that a molecule that is active will then go activate something else. It'll turn something else on. It'll change something else. And that's what we see happen in picture 3 here, down at the bottom. G protein, now that it's all energized and active and stuff, is going to mosey its way across the lip of the cell membrane here, and it's going to activate this enzyme right here. We notice this guy right here in picture number 2 is inactive, and then in picture number 3 it's active. 2 ways to figure that out. 2 ways to figure out that it's active. Number 1, it changes shape. Look at the shape of this guy here compared to the shape of this guy here. They're the same enzyme, but they have different shapes, right? And so what we see is that this inactive enzyme is now active because it has a conformational shape change, okay? The other indicator is that it's got this glowing yellow aura around it, right? We said that's what we want to see if a molecule is active, all right? So G protein right here, this green blob, is responsible for activating this guy right here, okay? Now let me erase all that. All right, and get rid of this too and this. All right. So what we see now is that this enzyme has been activated, and what do we see as the next step? A cellular response. Yes, that's a very, very generic way of telling you what happens next, but there's so many different cellular responses that the cell can have, we can't possibly go through all of them, and so they're just going to put that there for us to help understand what happens next, okay? Ultimately, there is a cellular response, and once this molecule here becomes activated, it will initiate the steps that will create or cause that cellular response, okay? Now, once this entire complex has been activated, it needs to reset, right? We want to be able to have G protein linked receptor, the purple guy there, accept another signal so that the cell can have another response, okay? So that's what we see happening right here. And in this final in this final picture, picture number 4. Everything else is resetting. It's going back to its beginning set stage, right? The enzyme here has gone back to its normal shape. G protein right here has kind of stopped glowing, okay? And then G protein linked receptor has kind of changed its shape back to normal as well. Okay, so it's resetting back to its default setting. And once it goes back to its default setting, it can take more signals and then have another response, and more signals and another response. Okay, so that's how G protein linked receptor works. The second type is called receptor tyrosine kinase. Okay? Now, the big difference between receptor tyrosine kinase and G protein linked receptor is that receptor tyrosine kinase can activate multiple pathways at once, okay? And it's a really nice little option to have because it can technically activate 6 pathways at once, 6 different ones, or it can do 1 pathway 6 times, or it can do 2 pathways 3 times, any combination, right? So that's what's really nice here about receptor tyrosine kinase, but it works in a distinctly different way, okay? So we're going to talk briefly about how this one works, and we'll be able to eventually compare it to G protein linked receptor as well, okay? So now, if we look up here, I'm just going to get my drawing tool out for this one as well. Look up here at picture number 1, okay? This is an entirely inactive receptor, okay? The receptor tyrosine kinase is the purple, the 2 purple things that we see embedded. Here's one of them, here's the other one. The 2 purple things that are embedded in the cell membrane. Now this one's kind of weird. It's got a little bit of a thing that hangs out at the top, okay, that goes out into the extracellular fluid. Oh, sorry, I'm covering up the camera. That's this thing right here, okay? And then you've got this big complex down here that hangs down in the middle of the cell. So it's distinctly different in the way it looks. Okay, so the next step, we're going to go ahead and start to activate receptor tyrosine kinase. And what we have to do to activate a receptor molecule in the cell membrane is to have it bind to a signal molecule. And so that's what we see here in picture number 2. Picture number 2 shows us signal molecules up here interacting with the active site of each of these receptors. Okay? Now, what do we know is characteristic of a molecule or a receptor that has some kind of interaction with the signal molecule? It is a change in shape. Okay, so if we notice here, there are two subcomponents, 1, 2, of this protein. Okay, and once molecules, signal molecules, bind to those two subcomponents, they will come together to form what is called a dimer. Okay, that's a generic term for two things coming together, a dimer. Now, I said that you would be looking for a shape change once the signal molecule has bound, right? Well, that's a shape change to me, right? I've got one thing and one thing. That's one shape, right? And then you bring them together, and that's a completely different shape, okay? It's a completely different conformation, right? And so that's what we see here. This is now going to become an activated molecule, and we see that down here. Now we see in picture number 3, if you look real closely, you can see that the receptor molecule down here is glowing yellow, okay? That's, again, how we know this thing is activated, okay? Now, the tyrosine kinases act a little bit differently than G protein-linked receptor, all right? They're going to use ATP to then activate other pathways, okay? Now, an activated tyrosine kinase, what it's going to do is it's going to break apart ATP, okay? And one of the things that you get when you break apart ATP, I'm going to pull this up here. Okay, ATP is adenosine with 2, 3 phosphates. Okay, linked to 3 phosphates. That's ATP, adenosine triphosphate. Now, when you chop off one of these phosphates, you can actually add it to something. Okay, so let's say this is a protein over here, a protein. You can chop that phosphate off and add it to the protein, and what you do is you activate that protein. Whatever happens to be, you turned it on. When you take one of those phosphates, you rip it off ATP, you add it to something else, that other thing is going to be activated. Okay, and that's kind of what we have happening here. Remember, we know as ATP is the energy molecule, and so that's a good way for you to remember it, that ATP is associated with these phosphates that you see right here. I'm going to go back to the slideshow real quick. These phosphates that you see right here, right by the pointer, okay, those are associated with energy, okay? And so you can think to yourself, okay, those phosphates are ultimately going to be used to activate something else, all right? So once the receptor tyrosine kinase is activated after it's changed its shape, it will break the phosphate off of 6 of these ATPs and attach that phosphate to itself, okay? And what this becomes is kind of like, if you like, spaceship dramas and things like that, like Star Wars and Star Trek and stuff like that. It almost looks like a spaceship docking, like a space station almost. And each one of these, let me get back to these things. Each one of these little areas right here, okay, there's 6 of them, 2, 3, 4, 5, 6. Each one of those little guys seems to me almost like an area where a spaceship could come and get some fuel, okay? And that fuel is the phosphate, okay? That high energy kind of molecule. And so the docking bays, okay, there's 6 of them, have fuel that little protein spaceships can come and take away, okay? And so once, that's what we kind of see going on here in the drawing number 4. These things even kind of look like spaceships. They are inactive proteins, okay? Here's one of them, here's another one, okay? And they will come flying in here to each one of these docking bays. They're going to grab one of these little phosphates that we see right here, and they are going to become activated as a result. And how do we know that? Let's look at the protein, okay? Compare this protein right here to this protein right here. Number 1 is changed shape, okay? So the fact that it went and got one of those little energy bundles, the phosphates, caused it to change its shape. And number 2, we see the little glowing red or yellow aura, right? So this inactive protein has turned on. It has become active. The same thing with this one. Again, look for shape change. This one has changed shape from here to here. Okay, changed shape. And we got the little glowing aura as well. Now, when you activate these inactive proteins that we see down here, they in turn are going to go activate something else. Okay, this top one is going to go activate cellular response number 1. This bottom one is going to go activate cellular response number 2. Okay, distinctly different responses, but both pathways have now been activated. Okay, so this is a way for, another way I should say, for our cells to take a chemical from the external environment and basically start a pathway inside of the cell, a signal pathway inside of the cell that's going to create some kind of cellular response. Okay, it's the second way. So now this one, as I mentioned before, is a little bit more flexible. It allows some flexibility in which pathways are going to be activated. It can activate one pathway 6 times, or 6 different pathways 1 time, or 3 different pathways 2 times, or 2, 3 times, whatever. You got all this different variability in different amounts of pathways that you can activate with this specific type. Okay. All right, almost done here. The final type of receptor we're going to talk about is called an ion channel receptor, and it acts as a gate when the receptor changes its shape. And this one's a little bit easier. This is probably the easiest of the 3 to really understand what's going on, because there's less moving parts. It's basically one moving part. Okay, and we can see that at the top here, this is an inactive receptor. It's called a ligand-gated ion channel receptor. Break that down. It's ligand-gated, which tells us it's going to open when a specific ligand interacts with the active site. Okay, this little area right here. Initially, it's got this little gate across the pore, all right? And that gate closes everything. Nothing's going to come through there. But when the signal molecule binds to this area, what it's going to cause is this little gate to flip up. Okay, and that's what we see in picture number 2. The gate has flipped up, and how do we know that that this thing has been activated? It has changed its shape. Okay, the gate is closed, the gate is open. I've never seen such an easy way to identify a shape change, right? And so what this does is it causes this channel, basically all this is now a hole in the cell, to allow certain ions through. Okay, so that's what we see, these green things. These ions were stuck out here in the first picture. Now we see them flooding into this inside the cell in the second picture. Okay, and this is going to elicit some kind of cellular response. All right, and when all of this stuff is done going through, this ligand will fall out of the active site and it'll close the gate. Okay, now we see this type of channel, gated channel, used all the time in our synapses. Okay, the connection between 2 different nerves. Okay, those gates that receive the signal at the second nerve will actually open due to something called a neurotransmitter. Okay, the neurotransmitter is a chemical that will open these ion channels. Okay, so that's where we see them in our body. All right, now there is a little animation here, and as far as I know, you should be able to see this. I'm going to go through the whole thing because it shows some of this stuff in motion. And when you start to see some of this stuff in motion, it starts to have a whole new life. You start to get an idea of exactly what the heck's going on. You're like, oh yeah, I get it now. I can see this stuff in motion. So I'm going to click through this stuff and watch, and you guys watch as it goes. And I'm not going to say anything over the guy. He's just going to talk. And hopefully you can hear it. And if not, let me know, and it's a talking point that we can touch on in one of our in-person sessions here, okay? All right, the reason I'm not going to, you can try to click it. I just don't think it'll work because it's a flash-based type of thing. Pardon me. It's a flash-based type of thing, and a lot of computers don't even run the flash stuff anymore. So let's see if this will work.
Most signal molecules act on target cells by binding to receptors in the cell membrane. There are several kinds of membrane receptors. Some signal molecules enter target cells and bind to intracellular receptors in the cytoplasm. 4 different kinds of receptors are shown here. Click on each receptor to study its function.
Okay, let's do the intracellular receptor. That was the first one that we had touched on, so we'll do that.
Some signal molecules actually enter target cells and act on intracellular receptors, proteins located inside the target cell. Steroid hormones like testosterone and estrogen act on intracellular receptors. Click on the signal molecule that interacts with this intracellular. Nonpolar molecules like steroids and thyroid hormones are able to pass through the plasma membrane and bind to a receptor protein in the cytoplasm or nucleus of a target cell. The activated receptor triggers a change in the cell. Steroids cause receptors to turn genes on and off. Click on another receptor type.
Let's do G protein linked receptor.
Many signal molecules, such as neurotransmitters and many hormones, act through G protein linked receptors. This kind of receptor spans the cell membrane, and as you might guess, it works through a protein called the G protein. Click on the correct signal molecule to activate the G protein link. When the signal molecule binds to the receptor becomes activated. It is now able to activate a specific G protein by causing GTP to displace GDP on the G protein. The activated G protein then binds to another protein, usually an enzyme, and alters its activity. Then the G protein hydrolyzes its GTP and reverts to its inactive form, ready to respond to another signal. Click on another receptor type.
Okay, receptor tyrosine kinase. Oh.
Growth factors signal cells to grow and divide by acting through receptor tyrosine kinases. Click on the correct signal molecule to activate this.
Binding of signal molecules causes 2 polypeptides to join, activating parts of each that act as tyrosine kinase enzymes, which then phosphorylate tyrosines in the tail of the other polypeptide. The receptor protein is now recognized by specific relay proteins inside the cell. One tyrosine kinase receptor may activate several relay proteins at once, triggering several different effects within the cell. Click on another receptor type. Some membrane receptors are ligand-gated ion channels. They are protein pores in the plasma membrane that allow or block the passage of certain ions through the membrane. For example, this occurs when one neuron signals another via a neurotransmitter. The signal molecule attaches to a site on the ion channel protein. This changes the shape of the protein, opening a channel through the membrane. Ions flow through the channel, and the change in ion concentration triggers a cellular response. Click on another receptor type.
Okay, so I hope you were able to see that. It really does put a lot of the stuff we just talked about into perspective when you can see it in motion. You may get the most benefit out of actually going back and reviewing the part that I just showed you. Hopefully it did work. Hopefully you can see that. If not, we'll try to show it live in class, okay? But as of this slide here, that does it for reception of signals. Now, the next section, the next video we're going to talk about discusses what happens with these arrows, okay? We're gonna get into specifics about what these arrows mean, okay? Because there's a lot more that goes on after we've received the signal. We've got to pass that signal through the cytoplasm of the cell, and we've got to monkey with it, we've got to change it, we've got to amplify, we've got to do all these crazy things to it so that we can get some kind of noticeable cellular response, okay? So I'm going to go ahead and make the third video, which is going to describe transduction. Thanks, guys.